Hello everybody, welcome back to another video. We are covering the antigens and humoral immune response. And my name is Bench Mariano. I hope you guys enjoy the video. I know it's uneven, you're all gonna say anything, but yeah, I hope you guys enjoy. So let's start off with this drawing of a B cell, which is a type of white blood cell. And the stems on the actual B cell have unique B cell receptors, or called BCRs, on the surface. Their job is to catch any foreign antigens to be displayed onto their stems, and through this, the receptor-mediated endocytosis, where the antigen is taken inside or basically in by the B cell. Alright, moving on, crumpling this piece of paper, we have a new model of a B cell to display the next step, where antigens are broken down and epitopes are presented at the surface along with MHC2 proteins. Furthermore, this little dot that's being shown on paper is the activation of the helper T cell. And what the helper T cell does is that it interacts with the B cell that has recognized the same antigen that entered the body. And it releases cytokines such as IL-2s to act on the B cell and eventually stimulate it. In the next process, the B cells would begin to divide rapidly in a process called clonal expansion, where many B cell clones would eventually become plasma cells while some become memory cells. These new plasma cells would eventually secrete large amount of antibodies that circulate through the blood and the lymph to get rid of the foreign object that came through. Okay, now bear with me. I know that was a lot to process, but for any of you that are confused, let's go for a simplified step-by-step process. So we have the first step, and the first step is that when a B cell is triggered, it encounters the matching antigen. And step 2, the B cell engulfs it and digests the actual antigen itself. And step 3, the antigen fragments are bound to unique MC2 molecules. And furthermore, moving on to a new paper, step 4, the combination of antigen and MCH molecules attracts the help of a mature matching T cell. And then moving on to step 5, where the cytokines are secreted by the T cell to help the B cell multiply and mature into antibody-producing plasma cells. And those plasma cells eventually secrete the antibodies shown on screen, and those antibodies are released into the blood to lock onto matching antigens to get rid of them. And that's it. Thanks for watching.